So how do we know evolution is actually occurring? Well, one tool that we use to distinguish whether or not allele frequency has changed is an equation known as the Hardy-Weinberg equilibrium equation, which essentially describes a population that is not evolving, where the alleles, from one generation to the next, the allele frequency stays the same. So, and I know this can look daunting at first, but it's really, once you get used to it, you'll see the logic of it, and it's actually pretty simple. So P is equal to the frequency of the dominant allele at a particular locus, and Q is the frequency, I don't know why I put a period there, is the frequency of the recessive allele. So not surprisingly, P plus Q is equal to 1 or 100% of the alleles in that population. So P squared is the frequency of individuals who are homozygous dominant. That makes sense, right? Because they have two dominant alleles, and we're calling the dominant allele P. Q squared is the frequency of homozygous recessive. Individuals. And 2PQ is the frequency of heterozygous individuals. So how this works is that if a population is in equilibrium, then both P and Q, the frequency of both P and Q, will stay the same in every generation. And so will P squared, Q squared, and 2PQ. So if a population is in Hardy-Weinberg equilibrium, the frequency of P and Q stay the same generation to generation. And I am going to have a separate video and or be talking in lab about how to actually apply this equation. Okay, we're not going to get into that in this lecture because this lecture is already so full of stuff. But we will be going over it. So that's just a little primer on it. I want to back up a slide. Sorry. I forgot to mention something. There are some conditions of Hardy-Weinberg equilibrium. So conditions of Hardy-Weinberg equilibrium. These conditions must be met for a population to be in Hardy-Weinberg equilibrium. Number 1, no mutation. Number 2, no natural selection. 3, extremely large population. Like approaching infinity large, okay? Because we can't have any genetic drift, okay? So that's kind of what's going on there. No drift. Okay. 4, no gene flow. And 5, it must be random mating. In other words, no sexual selection. You might be thinking, well, that seems pretty stringent. What population could possibly meet those criteria? Well, none of them, basically. Populations are always evolving. Hardy-Weinberg equilibrium never happens in nature. It is a baseline for comparison. So essentially, I mean, I can't say never, but more or less. In most populations, it's very rare, because one of those conditions will be violated. And you might have noticed, hey, those conditions look an awfully lot like my list of the mechanisms of evolution. Yes, because they are. So, yes. Anyway, we'll be calculating some Hardy-Weinberg in lab. It's going to be a good time. No, it will be, I promise. Okay, so I also want to go over some types of natural selection. So what I'm describing here is how the mode of selection influences the phenotype, the average or the most frequently observed phenotype in the population, right? Because that's what we see. Remember, phenotype is your physical appearance. So the three types I want to talk about, one is called stabilizing selection. So stabilizing selection, is where the environment selects against both extremes and favors an intermediate phenotype. Environment favors intermediate phenotype. So let's look at this example. So in this example, moths, I guess is what we're looking here, if they are too dark, they are selected against, maybe because a predator sees them better against the backdrop. If moths are too light, they are also selected against, maybe they don't get mates. And so this intermediate phenotype is actually selected for. Another really classic example of stabilizing selection is birth weight in humans. So babies that are too small don't have good survival. When babies are too large, it leads to complications, and survival is also compromised. And so there's been very strong stabilizing selection historically on human birth weight. Now medical technology is starting to change that a little bit, which is a good thing, but it's a good example of stabilizing selection. Directional selection, I feel like, is kind of the most easy to wrap your mind around. Directional selection is when one extreme phenotype is favored. In other words, we're going in one direction or the other. So in this case, we are having directional selection towards a lighter colored phenotype. But it could have been going the other way, and that still would have been considered directional selection. We're going in one direction towards one extreme or the other. And then in disruptive selection, the extremes sort of become more common, and the intermediate variation, or the intermediate phenotype, is selected against. So either extreme phenotype selected for, and intermediate selected against. So one example might be if you had, say you had a population of birds, and there were only very small seeds or very large seeds available. So birds with very little teeny beaks that could pick up the teeny seeds very easily did well, and birds with the large beaks that could crack the big large seeds did well, but birds with an intermediate sized beak could not get food, had poor survival, and didn't pass their genes on. That would be one example of what could cause disruptive selection. And disruptive selection is interesting because whereas stabilizing and directional, so stabilizing and directional tend to decrease variation in a population, disruptive actually tends to increase variation because we have more than one phenotype that is selected for. Okay, so I want to talk just briefly about a few other factors that can influence evolutionary change. These are things that Darwin didn't necessarily appreciate in his day, but we've come to realize can actually be very important in determining patterns of evolution. And the first is called frequency-dependent selection. Sometimes we just call this FDS. So this is a situation where the frequency determines your reproductive success. I guess that sounds pretty obvious, but so let me give you an example. So in this case, we're looking at some fish here. Now, these little guys right here, these fish, come in two different varieties. We have what's called the right-mouthed variety and the left-mouthed variety. Now, the right-mouthed variety, the way that the mouth of this individual is shaped, it can come up to this fish, and when it does, it takes a nice little bite out of it. That's what it does. So it has to be coming up on the left side of this fish. So what happens is if there are a lot, or if in other words, the frequency of this right-mouthed fish is very high, then the bigger fish get good at fending them off. And so they become selected against. And the fish that will have the advantage then are the left-mouthed ones, because they're not going to see them coming, right? This left-mouthed one takes a bite out of the right side of the fish. And so this guy is going to start to become favored in terms of, passing its genes on, because it's going to have a greater opportunity to get a meal and not get killed. So with frequency-dependent selection, typically the less frequent morph in the population has a selective advantage, and thus increases in frequency until it loses its disadvantage. And then, again, the less common morph will be advantage. So, that wasn't a real sentence. Will be advantage. What I mean is, so this left mouth might have the advantage when it's rare. And that will cause it to proliferate and become more and more common. Once it reaches a threshold where it becomes too common, though, then the advantage switches. And the right-mouthed ones will have the advantage. Okay, so that is frequency-dependent selection. How successful you are depends on the frequency of your phenotype in the population. I'm actually going to write that. How successful you are depends on your frequency in the population. And of course, by your, I mean your genes, your phenotype, which there is, I'm talking about phenotypes a lot, but of course recognize that there are genes responsible for that phenotype. Another interesting thing that can control patterns of evolution is something called heterozygote advantage. And this is where heterozygotes are more successful than either homozygote. Now this will tend to increase variation because we're keeping both those alleles are doing well. So, oops, I'm ahead of myself. So with heterozygote advantage, one example we're looking at here are a genus of butterflies known as Coleus butterflies. And these butterflies have a gene that produces a protein that influences their ability to fly at different temperatures. There is an allele for high temperature and an allele for low temperature. Those who have each allele, in other words, are heterozygous, survive better at a broader range of temperatures than either homozygote. So that would be an example of heterozygote advantage. There is also another factor in play, basically the environment. So geographic variation is also very important in influencing evolution. Different environmental conditions will favor different phenotypes. So one example is seen in white clover. So white clover can produce cyanide to protect itself against herbivores. But one problem is that when the plant experiences a freezing temperature, as it would in some regions, it causes the cyanide to be released to the plant's own tissue, which will kill the plant. So that's not good. That's not going to help you pass your genes on at all. So what we see is this kind of geographic variation, and so the red is showing the having the allele to produce cyanide. As we go into temperatures, as we have geographic variation of colder temperature, we see a shift in allele frequency. Because in different environments, different gene combinations will be more or less successful. So environment has a really profound influence. And then finally, I want to talk a little bit about just some limitations of natural selection, because you might think, well, how come people can't fly then? Because wouldn't that be adaptive? Well, it doesn't work that way. Mutations are random. You don't get a mutation because you need one. And for starters, selection can only act on existing variation. You have to have the mutation first, okay? You have to have the mutation first. Also, every organism is limited by historical constraint. In other words, they're bound by what their ancestors had to work with. Okay, so for example, we're looking at a couple of different organisms. One is a bony fish, and one is a ray with a cartilage skeleton, and they both are have adapted to be sort of bottom dwelling and sort of just kind of be very, very flat against the bottom of the earth. But because of their very different skeletons, they have had to evolve different strategies to achieve the same sort of environmental goal, right? They have different things to work with. Think about a bird's wings. Now, it would probably be pretty handy if the bird also had some, paws in the front to grab stuff with, but the forelimb of its ancestors evolved into a wing. So, sorry, those, you can't have paws because they became wings. So, you're constrained by what your ancestors kind of gave you. And then also adaptations are often compromises. That's not spelled right, sorry. So, looking at this example here, so we have this little newt who's not doing so well in this picture, but this newt is really cool. It produces a neurotoxin, however, which of course is to keep predators from eating it, but there is this kind of garter snake that can eat this newt and not die. The garter snake will be immobile for several hours themselves, though, after eating the newt, and that makes them vulnerable to predators. So, it's a compromise. They can eat this toxic newt, but they then themselves are more vulnerable to predators. So it's a compromise, in nature. It's wild out there, man. Okay, so that's it. Please let me know if you have any questions about this or anything else, and I will do my best to address them, and I will see you guys in lab.